This is Chapter 2, Lecture K, Nucleic Acids. And on your screen you see two of the most important nucleic acids, DNA and RNA. And it's important that you know some of the differences between DNA. Two of the significant differences are shown here. First of all, in DNA, it contains the nitrogen base thymine, whereas instead of thymine, RNA contains uracil. The other significant difference is that, as the name implies, DNA has a sugar called deoxyribose, whereas RNA has a sugar called ribose. Some of the other significant differences are that DNA is only found in the control center of the cell, and that control center of the cell is the nucleus. The DNA is our so-called blueprint of life. It contains all of our genes that are necessary to make various proteins. The RNA is only going to be found in the cytosol, and it's going to be important for acting as a copy of the DNA. The other important difference is that RNA is single-stranded, whereas DNA is double-stranded, and it forms a double helix. So DNA stands for deoxyribonucleic acid, and RNA stands for ribonucleic acid. It's the largest molecules in the body, and as a group, nucleic acids are the largest molecules in the body. Very, very significant. Nucleic acids contain carbon, oxygen, hydrogen, nitrogen, and also phosphorus. Now, the monomers of nucleic acids are referred to as nucleotides, and these nucleotides contain the nitrogen base, a pentose sugar, and also a phosphate group. So in the case of DNA, there are 4 different nitrogenous bases, nitrogen bases, and 2 of them are called adenine and guanine. And both of these are classified as purines. And one of the reasons they're called purines is because of their structure. If we go back to our first slide here, you can see that adenine and guanine both have a double ring structure. And we can see some similarities between cytosine and thymine as well. So as you can see, cytosine and thymine here are classified as pyrimidines. And the beauty of this, of the size of these particular purines or pyrimidines, allows them to form this double helix by the way that they bind with each other. And a purine is always going to bind with a pyrimidine in a double ring structure to form this double helix. So for example, the adenine is always going to pair with thymine, and it's going to be connected by hydrogen bonds. And there's always 2 hydrogen bonds between an adenine and a thymine. The hydrogen bonds are going to be important in stabilizing this double helix. Then we always are going to see cytosine pairing with guanine. And in this case, there's always going to be 3 hydrogen bonds between a cytosine and a guanine. So these are really the DNA molecule would be the ultimate instructions for protein synthesis. It is all the directions to make the different proteins that we need, and it's very crucial that the DNA never be damaged so it never leaves the nucleus. This picture is figure 2.22 that you have in your textbook, and it's important for you to know how they pair. So again, adenine is always going to pair with thymine. So a good way to remember this is I always remember North Carolina A&T as a college in Greensboro, North Carolina, and then G always pairs up with C. And there's another school there called Greensboro College. These are all little memory tricks that will help you, little mnemonics, and hopefully this will help you remember that G pairs with C, just like A pairs with T. So in the test, if I were to give you an example of a sequence of nucleotides, for example, A, TA, TA, G, 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 since you know what nucleotide pairs up with the other one, you should be able to tell me which ones pair up with these. So the complementary sequence of DNA would be A, T, A, TCCG. So RNA is going to be single-stranded. So remember, DNA is double-stranded, RNA is single-stranded. It has adenine and guanine cytosine, but again, instead of thymine, it has uracil. So this is one of the big differences. And there's 3 different types of RNA you should be aware of. The first type is called messenger RNA. And messenger RNA is going to be an exact copy of the DNA. There's also transfer RNA, which is going to be used for translation or the ability to actually synthesize a protein. So this would be protein synthesis. The other type of RNA is called rRNA, ribosomal RNA, and this is the structural component of ribosomes. The structural component of ribosomes. And again, there's an important animation that you need to watch that's in Mastering AMP here, DNA and RNA. Now when we think about nucleic acids, we also have another very common nucleic acid, which would be ATP, adenosine triphosphate. And ATP, adenosine triphosphate, is really the currency of the cell. It's the currency that is used for all transactions that happen. So if a reaction needs to happen, if anything needs to happen in the body, ATP is needed to do that. In order for us to contract our muscles, ATP is necessary to do this. So you should be aware that ATP has adenine. This is the same adenine that's found in DNA. It has a sugar called the ribose sugar, so not deoxyribose, but just ribose. And then it has these phosphate groups. You can see there's 1, 2, and 3 phosphate groups. So when there's 3 phosphate groups, it's called ATP. When there's 2 phosphate groups, it's ADP. And when there's 1 phosphate group, it's called AMP for the monophosphate. And the phosphate groups are really the most important part of the ATP molecule, because when these bonds that I'm pointing to on the screen are broken, this causes energy to be released. So it releases energy, and this energy is used for a reaction in the body. So it releases energy, which is used for reactions in the body. And we'll be talking a lot about ATP all throughout not only this semester, but you'll have it in the next semester as well. This slide is showing 3 different examples of cellular work driven by the energy that is actually released from ATP. And this is figure 2.24 in your textbook. In the first example here we have transport work. So whenever any molecule has to go across the plasma membrane, ATP is going to be necessary for that to happen. So essentially what the ATP does, there's a chemical reaction where ATP is broken down into ADP, and that releases this phosphate. And so again, energy is released, which allows this protein pump to actually open, so that now a single molecule can exit the cell. And there's an enzyme that carries out this reaction. This enzyme is called ATPase. ATPase. Remember, you've learned that one of the characteristics of enzymes is that oftentimes they will end with ase. So an ATPase is going to break ATP down into ADP. Another example of how ATP is used in our body, we're going to talk about this when we get to chapter 9. We have a relaxed smooth muscle cell here. In order for it to contract, ATP is going to be released, and that allows for this muscle to contract. So again, ATP is used to do work in the body. A third example is chemical work. So in order for A and B to join, this would be an example of a synthesis reaction. In order for this reaction to happen, ATP is also going to be present.